Hello, today we're going to discuss protein synthesis. So, what is protein synthesis? It is the process by which amino acids are linearly arranged into proteins through the involvement of ribosomal RNA, transfer RNA, messenger RNA, and various enzymes. The instructions for making proteins are genes. So a distinct sequence of nucleotides forming part of a chromosome, the order of which determines the order of monomers in a polypeptide or nucleic acid molecule which a cell or virus may synthesize. So, there are three main parts of protein synthesis. There is transcription, RNA splicing, and translation. Now, before we can even begin transcription, we need to learn about the promoter, operator, and terminator sequences. So in order for the RNA polymerase to know where to begin and end transcription, it needs to read promoter and terminator sequences of DNA. Sometimes the promoter sequence occurs as TATAAA and is nicknamed the TATA box. Those T's and A's, again, they stand for thymines and adenines. Now, the key difference between the promoter and the operator is based on the type of molecule that binds to the respective DNA sequence. RNA polymerase binds the promoter, whereas regulatory molecules of the operon system bind to the operator. Both are essential to, again, starting transcription. Now, transcription itself, I'll just start with the definition. So it's the process by which genetic information represented by a sequence of DNA nucleotides is copied into newly synthesized molecules of RNA, with the DNA serving as a template. So this process occurs by RNA polymerase unwinding a section of DNA and making complementary RNA nucleotides to the DNA nucleotides read. RNA polymerase reads the DNA going in the 3' to 5' direction. RNA polymerase synthesizes RNA nucleotides going in the 5' to 3' direction, and this happens entirely in the nucleus of eukaryotes. Now, RNA splicing. So the RNA synthesized by the RNA polymerase is actually known as pre-mRNA. For it to become mRNA, it must be spliced. So what is RNA splicing? It is a process in molecular biology where a newly made precursor messenger RNA transcript is transformed into a mature messenger RNA. It works by removing introns and so joining together exons. So introns mean intervening sequences and exons mean expressed sequences. So you only want the express sequences, you don't want any of the intervening sequences. So this is how you can properly make a protein, which is going to be properly expressing whatever the gene is coding for in the DNA. So the way splicing works is by a spliceosome. So a spliceosome is a ribonucleoprotein complex. That is the site in the cell nucleus where introns are excised from precursor messenger RNA and exons are joined together to form functional messenger RNA. All of this happens entirely in the nucleus of eukaryotes. So here is what RNA splicing looks like. So if we look at the GIF, you can see the spliceosome protein complex. It basically finds each end of the intron, folds and kind of cuts out the intron while simultaneously joining the exons together. The image on the left, you can see how that is joined together without the spliceosomes. Okay, translation now. So after that splicing, we now have translation. So I'll start with the definition. In biology, it is the process by which a sequence of nucleotide triplets in a messenger RNA molecule gives rise to a specific sequence of amino acids during synthesis of a polypeptide or protein. So, translation is happening in cytoplasm. So, mRNA strand moves into the cytoplasm where it meets up with the ribosome. Ribosome recognizes the start codon and begins translation. Start codon is different than the promoter and operator sequences. The tRNA carrying amino acids have complementary anticodons to the mRNA codons being read by the ribosome. This translation occurs in the 5' to 3' direction. When a tRNA molecule has complementarily matched with the mRNA sequence, the amino acid is dropped off and used as part of a protein. This occurs until the ribosome recognizes the stop codon. Again, different from the terminator sequence. This all again happens entirely in the cytoplasm of eukaryotes. Okay, this is the last slide, and it kind of summarizes everything we've talked about. The bottom right GIF is going to be showing transcription and translation, but it doesn't show the splicing, which is why I again have that image in the GIF in the bottom left. And it also, starting with, I guess, our promoter and operator sequences and terminator sequences, that also is a transcription step that is not shown in that bottom right GIF, but it still helps to kind of piece everything together. So I'm going to let this GIF in the bottom right play through because it's kind of lengthy, and then that will be it for this lesson.
Thank you for watching. Please share and subscribe, and remember to spread science.